Hello friends, today we are going to discuss one of the important sub-theme in Biomolecules chapter, DNA damages and repair mechanisms. DNA damages and repair mechanisms. We know genes present in DNA are the crux point for physical and physiological effects. Therefore, proper maintenance of DNA replication is essential for the continuity of heredity. Any error or any mismatch during DNA replication leads to adversarial consequences simply called mutations. That means the DNA replication, if there is a mismatch during DNA replication, leads to mutations. That means the entire survival or the existence of life depends on this DNA replication. Before we go into the exact topic, we have to discuss about some of the key issues. There are 10 to the power of 14 nucleated cells on an average in a human being. Nucleated cells. And there are 3 into 10 to the power of 9 base pairs per cell. And there are 10 to the power of 16 cell cycles in a lifetime. I told you, there should not be any error during replication. And what is the rate of that error? Possible rate of error on average? It is 1 error per billion base pairs. I told you, this mismatch or this leads to mutations which is unwanted company. And what is that rate? It is 1 mutation per 10 to the power of 6 billion base pairs. This shows that DNA mismatches during replication is at very least proportion. Not too often, we can say, because we have effective DNA damage repair mechanism, for sure. But the DNA replication mismatches, what we call errors, that leads to mutations, do occur in all nucleated cells. So today we are going to discuss about that and what are different types of DNA damages and what are its consequences and how these DNA damages has been effectively repaired by our repair mechanisms and what are its types and how it leads to different cancers. For example, HNPCC cancer because of a mismatch repair. That means because of a mismatch. So, and to avoid the same mismatch repairs comes into existence. Then we will discuss point by point, okay? And with regard to different DNA different damages, there are, depending on the kind or the intensity, or who cause exactly the DNA damages. There are, say, physical agents, the chemical agents, the environmental agents, like for example radiation, the physical, chemical, environmental agents cause DNA damages. For example, radiation, chemicals, what we call simply mutagens. That means apart from mismatches during DNA replication, these are the causatives for DNA damages, which all together leads to mutations. Mutations. Broadly, depending on the kind of agents and other factors, there are different kind of DNA damages. For example, single base alteration. Single base alterations. Say depurination, deamination. This is very important because cytosine become uracil because of deamination. Adenine become hypoxanthine because of deamination. Guanine become xanthine because of deamination. And base alkylations, incorporation of base analogues. There are different single base alterations, then two base alterations. Say for instance, pyrimidine dimers. Pyrimidine dimers. Because of covalent linkages. How they form? Because of radiations. Say UV radiation may cause to form pyrimidine dimer. For example, TT, in the way of a formation of a covalent linkage. Simply TT dimers. Say for instance, chain break. Another kind of DNA dimer, chain breaks. Then cross linkages. This chain breaks is because of oxidizing free radical formations. This is very important, and they increase by age. Cross linkages within the same strand or opposite strand or between DNA and protein. So these are the different DNA damages. Single base alterations, two base alterations, chain breaks, and cross linkages. Single base alterations are very important because there are different types. Depyrination, most important, deamination. Because, for example, a cytosine becomes uracil, and we will have to discuss at this point at length when compared to others. Okay, adenine becomes hypoxanthine and G becomes xanthine. Base alkylation because of alkylations. Analogs, unwanted analogs may cause chain breaks. Chain breaks simply cause a free radical attack. Increasing in range, leads to free radical attacks. At the same time, oxidations, even radiations, which ultimately leads to chain breaks, is one of the DNA damages. Then cross linkages, maybe within the same strand or between opposite strand or between DNA and a protein molecule. So look at this point. How a cytosine becomes uracil? I told you already, because of deamination. Simply because of deamination. We all knew that, say, 2-oxy 4-aminopyrimidine. 2-oxy 4-aminopyrimidine. The cytosine. Adenine, we knew that 6-aminopurine. 6-aminopurine for adenine. And guanine, 2-amino 6-oxypurine. 2-amino 6-oxypurine. This deamination, this removal of amine groups, converts a cytosine into an unusual bases which are not usually present in DNA. For example, cytosine becomes uracil and adenine become hypoxanthine because of this deamination, whereas guanine becomes xanthine. So this is how the deamination works. Then we already discussed that all these DNA damages leads to mutations. So what are mutations? It is very important. What are mutations? A sudden change in the DNA structure of a gene. A sudden change. And there are two kinds. One, point mutations. Two, frame shift mutations. Point mutations are again of two kinds. Transition and transversion. Now, ultimately this leads to different kinds like silent mutations, missense mutations, and nonsense mutations. What is a transition? First, before that, what is a point mutation? A change, a mutation happening in a single base pair. just single base pair, but the thing is it happens in genes, because we knew that DNA contains lot of junk nucleotide sequences. As per the Human Genome Projects, around 98% of the DNA is a junk DNA. Though there is certain importance in the way of gene regulation or the regular gene function, but the majority or the most part of the DNA is junk. So therefore replication mismatches during this part won't affect. But the thing is, if it happens in gene bearing portions, this cause effect. Just a single base pair is enough that leads to adversarial consequence, what we call point mutations. And based on the kind of mutation, say whether, say in transition, a purine is replaced by another purine, whereas at the same time a pyrimidine replaced by another pyrimidine. That means A is a purine replaced by guanine, another purine. Say C is a pyrimidine replaced by another pyrimidine, thymine. Whereas transversion is different. One purine is replaced by another pyrimidine. One purine by another pyrimidine and one pyrimidine by another purine. This is the opposite. That means one purine replaced by one pyrimidine or vice versa. Say there is a sickle cell anemia. The most important transversion mutation. The sickle cell. We will discuss about this later. First, see, these are the basics, and what is a silent mutation? This is very important. For example, we knew that the information in DNA shall be copied into an intermediary complementary mRNA. That means genetic code in mRNA, and the same shall be translated in the way of a polypeptide chain, what we call transcription and translation. Central dogma of life. We knew that. And what are silent mutations? In that sense, silent mutations means there are different genetic codes. AUG for methionine, for instance. If a mismatched codon encodes a same amino acid, it is called silent mutation, because it doesn't cause any effect in the protein chain synthesis. It doesn't cause any physiological effect because the same amino acid occurs. Whereas missense, the name it implied, if the mismatched genetic code encodes a different amino acid which have an adversarial consequences, what we call missense mutation. And nonsense, if the genetic code leads to a terminator codon, that means UAA, UAG or UGA. If that means these terminator codon signals to a termination of a polypeptide chains. That means premature termination of polypeptide chains is a consequence of nonsense codons. That means nonsense mutations. Silent mutation, for example, we will elucidate the same with respect to an example. Say UCA codes for serine. Okay, UCU. It leads to another codon because of a mutation in the third base of a codon. It also encodes same amino acid, serine. Take another issue, ACA. That means first base in a codon has been mutated. This codes for threonine. Then take another issue, UAA. That means terminator codon because of a mutation in the second base. That means UCA is a genetic code that codes for a amino acid serine, whereas UCU is another genetic code that codes for serine. The thing is, though there is a change in the genetic code, but the amino acid is same. There is no change. But the thing is, there is another concept here. U has been replaced. U has been replaced by A. It becomes threonine. That means missense mutation. Then another codon, that means second base, UCA become UAA, terminator codon, leads to nonsense mutation. So that's how the issue ends. Say based on the kind of mutation, the consequence will come. Okay. Then These are about different things, and we will tell you how transfusion occurs. That means with respect to sickle cell anemia. That means, say transfusion. What exactly it is, we've already discussed. It's one of the important mutation. Say the classical example is sickle cell anemia. Sickle cell anemia. Say we knew that our hemoglobin is made up of 4 different polypeptide chains. Alpha 1, alpha 2, beta 1, beta 2. In the beta hemoglobin chain, that means in turn, because in the beta hemoglobin gene, at 6th position there is a transfusion mutation, because of which thymine has been substituted for adenine. How? For example, GAG. It codes for glutamic acid. Take another, it becomes GTG. How? For example, GAG means CTC is normal. In DNA I am saying. Okay, this is mRNA. And this has been replaced by CAC. That means transversion. It is at 6th position in the gene, in beta gene, beta hemoglobin gene. It is normal, it is abnormal. Thymine, which is a pyrimidine, has been replaced by adenine. That means a purine, that is transversion. One pyrimidine replaced by another purine, and it leads to conversion of a healthy codon into an abnormal. This is healthy. That means normal, because normal codes for glutamic acid, and this become abnormal, that codes for valine. Abnormal codon. Therefore, sickle cell shaped hemoglobin comes. Because of a single base pair. I told you already. If it is in junk DNA, no need to worry. If it is happening in a gene bearing position of DNA at the rate of 1 base pair, it is enough. That leads to adversarial consequence, wherein sickle cell anemia is a terrific example. Just thymine, which is a pyrimidine, replaced by adenine. That means T is substituted for A, transversion, and become GAG become GTG. That means glutamic acid replaced by valine. In valine, then sickle cell anemia. It leads to. That is the impact of mutation. I told you already, mutations are sudden, random, discontinuous, and unwanted, of course. Then, to counter these, we have repair mechanism. So it's about different consequences of DNA damages because of mutations, and how would we counter? We have a repair mechanism. We have an effective repair mechanism. Repair mechanisms. Say first, base excision. Base excision repair. Nucleotide excision repair. Mismatch repair. Double strand breakup. Base excision. What is excision? In general, excision means surgical removal. The same. Base excision means removal of an unwanted base. Nucleotide excision means removal of an unwanted nucleotide. Mismatch. Removal of unwanted that means unwanted mismatches. That's how the excision means, okay? And these are all very important. There are different kinds of repair mechanisms. We'll discuss one by one in the way of, that means by illustrating a diagram for better conduciveness. No? The first one, base excision repairs. Base excision repairs. We have discussed that depurination, that's what deamination simply, that leads to C become uracil, A become hypoxanthine, and that means adenine becomes hypoxanthine, guanine becomes xanthine. How? For example, say it's a normal DNA. This is normal. Because of deamination, cytosine becomes uracil. See. This is important. This is an unwanted thing, you know. And how it should repair. Because of this, the first action, uracil DNA glycosylase. It will remove this. That leads to A, T. Then endonucleases. This entire part has been cut down. Then DNA polymerase and DNA ligase replaces the healthy sequences, you know. This is normal. So this is how the sequential representation. Normal because of deamination, cytosine becomes uracil, and because of glycosylase action, it has been removed. Then it will lead to endonuclease activity. That entire segment has been removed. Then DNA polymerase add the new nucleotides, whereas DNA ligase esterification. That's how repaired. Done. This is about base excision repair. You know, next one, nucleotide excision repairs. Nucleotide excision repair. You know, sometimes because of ionizing radiations, say UV radiations, it causes, you know, modification of bases. Even the DNA strand will break up. So they have long-term effect, and these large-scale damages is effectively repaired by nucleotide excision repairs. How? For example, this is a normal DNA. So these are defected bases. Okay, defected part. Step 1. Step 2. Identifying the defected part and unwinding. This is very important. Then excision nucleases, what we call, they are very important. Excision nuclease, what exactly they will do? They will remove the damaged part. Then resynthesis, relegation, because, you know, we knew that DNA polymerase add nucleotides and DNA ligase align it. And the defected part will be degraded. How? That means here the thing is how they have to identify the defected part and unwinding, then the action of excision nucleases they will trim out, then the next action, that means resynthesis and relegation. The degradation of defected part will take place, followed by the DNA polymerase activity that which acts on healthy nucleotides, and DNA ligands which are what will form esterification, formation of ester bonds. That's how the resynthesis, what is simply called resynthesis and relegation. So it's about nucleotide excision repair. Here, identification and unwinding is very important, and this activity. Okay. Then mismatch repair. Mismatch repair. We have discussed during replication process, one base pair, simply one base may erroneously add into the daughter DNA, and how it get rectified because of this mismatch. How? For example, now the parental DNA generally is in methylated form. The parental DNA generally is in methylated form, whereas the daughter DNA is in non-methylated form, and this is a defected part. That means mismatched base pair. This is a mismatched base pair. There is an activity of an enzyme called GATC endonucleases. GATC endonucleases. And how it will done? The GATC endonucleases will cut down a specific fragment adjacent to the methylated bases. Adjacent to the methylated bases. Adjacent to the methylated bases. Please for a while. Adjacent to the methylated bases. Then the third step, the exact part, that means the entire part will be removed by exonuclease activity, then followed by the final and most important point, the DNA synthesis and relegation. DNA synthesis and relegation is very important. That leads to the restoration of healthy DNA. That means because of DNA polymerase and DNA ligase. That means the GATC endonuclease will cut down a specific fragment or adjacent to the methylated bases. Then the followed by the exonuclease activity that will remove the damaged sequence, that means then the DNA polymerase and DNA ligase. This is how the mismatched base pair will be restored. And the last one is the double strands. This is very simple. Double strand base breakup. By means of homologous recombination, it will get replaced. So these are about different DNA repair mechanisms. Okay, you can like, comment, and share. Thanks for watching.